Look, I'll start revision over here, and in case if you have like I have dictated each and everything, so I'll start from your PPT only. So then, in case if you have any doubt, any problem with that, then you can like pause me over there, and right. So then you will pause me over there, and you can reconfirm or clear your doubt over there only. Fine. So first, what is the transport? Look, here I am going to explain. Look, transport first, the transport between the cells, right? So how does the transport between the cell takes place? So we have studied that when the transport takes place between two cells, that takes place through with the help of a small pore-like structure, and these pore-like structures are known as plasmodesmata. So two cells remains connected with each other with the help of plasmodesmata. And look, the part over here, like look, this part over here, this is the protoplasm, which is peripheral, right? And here in the same way, the part which is like over here, this is the nucleus. So nucleus and the protoplasm both are peripheral in a plant, right? Why? Because vacuole takes a larger size, and vacuole pushes the protoplasm and the nucleus, right? So these are the pores which are known as the plasmodesmata. Translocation of the food and translocation of the mineral, translocation of the water, all these content take place with the help of these pores, which is called plasmodesmata, right? So what is the means of transport? So that is the transport within the short distances, right? This is the short distance transport, right? This short distance transport takes place by means of two things: diffusion and osmosis. And with the help of diffusion and osmosis, the transport of mineral, transport of food, transport of water takes place from one cell to another cell with the help of plasmodesmata, right? Now, when we talk about the translocation for the long distance, so here we are talking about the diffusion process, right? Here we are talking about those plants where transport also takes place in those plants where xylem and phloem-like structures are not formed. So, like the algae, like the bryophyta, they do not have xylem and phloem kind of the tissues. So, transport in those lower plant takes place with the help of diffusion and osmosis, right? Now, what is the long distance transport and how does the long distance transport takes place? Look, the long distance transport takes place with the help of xylem and phloem, right? Look, how does the long distance transport takes place? Now, let me explain you how does it take place. Look, in long distance transport, with it takes place with the help of xylem and phloem, right? In case of the xylem, what happened? In case of the xylem, uh, water transport takes place. Where? From the root to the leaves, right? So these blue color lines are showing the xylem. Xylem element are made up of four kind of the tissues. We have already understood, we have already studied it: tracheid, vessels, xylem parenchyma, and xylem fiber, right? And how does the transport? The transportation takes place through the transpiration pull theory. We have already studied and we will revise again, right? So what happened in xylem? Movement of the water takes place along with the movement of water and mineral, in fact. So movement of water and mineral takes place by the process of transpiration pull theory, right? And this is the long distance transport of the water, right? Again, so transpiration takes place at the leaf. It pulls the water where? From the root, and root absorb the water from the soil. So that's how the long distance transport of the water takes place, right? Now what about the food? Formation of food like glucose takes place here in the leaves with the help of photosynthesis, right? But glucose is not transported. Transportation of sucrose takes place. Sucrose is transported, right? So what happened? Glucose is converted into sucrose, then it is transported to those parts where food formation is not taking place. Where? Root. It is translocated toward the root, toward the food, multi-directional, right? So phloem help in the translocation of organic substances like food, right? Phloem is made up of tube cells, companion cell, phloem parenchyma, and phloem fiber. And we have already understood that translocation, long distance translocation of the food takes place with the help of mass flow hypothesis. We will revise it again as well, right? So in the nutshell, what I want to say you, that is the complete scenario of this complete chapter. This is the summary of complete chapter. Translocation takes place in short distance with the help of plasmodesmata through diffusion and osmosis. Transport of substance in long distances takes place with the help of xylem and phloem, and in the transport of water, transpiration pull theory work, and in the transportation of organic food, mass flow hypothesis work, right? Is it clear? Is this concept clear to all of you? Keep on telling me. Yes, all of you. Shakullah, Ishra, and Shahid.
Yes, sir.
Keep on asking, right? Keep on asking and keep on telling me that you are getting the right. So clear to all of you. Fine. So this was the summary of the transportation chapter which we are. So first we will study about the short distance transport. Look. Look. Now the means of transport, right? What are the means of transport? This is very important. It's very, very important. So what are the means of transport? How many means are used in the transport naturally, right? So transport is of basically two types, right? First, we have studied that active transport, and second, we have studied that is the passive transport, right? In active transport, the movement takes place where uphill. That is the against the concentration gradient. That is the opposite to the diffusion gradient. What is the opposite to the diffusion gradient? From the low to high concentration. So movement of the substances takes place from low concentration to high concentration. That is the opposite to diffusion gradient. That is the uphill, right? So in active transport, the energy is spent. So what is the form of energy? ATP is spent. So ATP is spent over here. At the expense of the ATP, diffusion of substance takes place against the diffusion gradient, right? Now the transport, passive transport. Passive transport takes place with a downhill, right? So passive transport takes place in downhill manner, right? What is the downhill manner? Downhill means whenever you come the downhill, so you need not to put much more energy to your cycle, your car, right? In the same way, downhill means that takes place towards the direction of diffusion gradient, right? That is from high concentration movement takes place from high concentration to low concentration. And the third most important thing, energy is not required in passive transport. So this is the active transport and this is the passive transport, right? Now, what is the passive transport, right? So passive transport takes place by the two means. Now, first, the diffusion through the lipid bilayer, right? Diffusion through the lipid bilayer. So lipid soluble substances, they are hydrophobic, right? Right? They are means they are the fat soluble substances. So fat soluble substances, they are the transported with the help of they are transported through the this bilayer of lipid, right? So bilayer of lipid, how does like this way? So this is the plasma membrane. Here I have made this bilayer of the lipid. These are the single lipid molecule, right? These are the lipid molecule, right? These are the lipid molecule, and out of these lipid molecule, their hydrophobic head, it always remains. So the lipid molecule looks like this one. This is their head and this is their tail, and this upper part which is the head, This head is hydrophilic. Head is hydrophilic, right? And this is their tail. Tail is hydrophobic, right? This is the hydrophobic, right? Hydrophilic and
So like means hydrophilic means like it's soluble in water.
yes, soluble in water. Means hydro means water and philic means loving. So those which are water soluble, right? Means water loving. Actually, this is Latin word. Hydrophilic means water loving. Water loving means those which are soluble to water, right? And hydrophobic, they are water repelling, right? Means they are fat soluble. That is water repelling. That is fat soluble. Clear? So those which are the fat soluble substances, they pass through with the help of diffusion. They pass through this bilayer of lipid, right? So they are lipid soluble substances. They directly pass through. But those which are the hydrophilic substances, means those which are the water soluble substances, they do not pass through this lipid bilayer. Now, how they will come inside? So diffusion through the protein. So diffusion of those substances takes place with the help of these proteins. These proteins are called channel protein, right, or carrier protein. So with the help of channel protein, right, they transport it from higher concentration toward the lower concentration. So in the passive way, diffusion takes place with the help of two things, that is the diffusion through the lipid and diffusion through the protein, right? Now tell me, is it clear or not?
Yes, sir.
Clear to all of you? Okay. Then come to the channel protein. Channel protein are of two types. Those protein which transport water and water soluble substances, they are called aquaporins, right? So channel proteins are two types. Those which transport the water, they are water soluble substances, hydrophilic substances, they are called aquaporin. And those which transport the ion, they are called ionic channel, right? So channel proteins are of two types, aquaporin and ionic channel. Now channel protein, right? So channel proteins, again, when we talk about the channel protein, they with the help of channel protein, simple diffusion takes place and facilitated diffusion takes place. Both are the passive process. In simple diffusion, this is the simple diffusion, just a substance is crossing from here to here, right? It's coming over here. For the facilitated diffusion, there is a special type of the protein in channel, that is called carrier protein. So carrier protein, it takes the substrate at one end and deliver at the another end. So that is the, so channel protein may be of two types, water, that is aquaporin and ionic channel. And with the help of these channel proteins, diffusion takes place with the help of simple diffusion or facilitated diffusion. In facilitated diffusion, carrier protein is not required. In simple diffusion, carrier protein, in facilitated diffusion, carrier protein is required, and in simple diffusion, carrier protein is not required. Tell me, is it clear or not?
Yes, sir.
Safla, clear?
Yes, sir, clear.
Shahid, all clear? Because this is the basic, this is the main concept of this chapter. Very good, very good. So feel free to ask me wherever you have any doubts, right? Now come to the passive transport, right? And active transport. Look. In passive transport, I have already told you that this is the diagram, which is well explained diagram. Fat soluble substances, they comes through the bilayer of the lipid with the help of diffusion, and it is a passive transport. In facilitated diffusion, right, they comes to the, this is also passive transport. They both of they takes place in the direction of diffusion gradient, but the carrier protein is required. In active transport, energy is, right, spent, right? And the protein or the channel protein which are required in active transport, these channel proteins are known as pump, right? Look, now the type of channel protein, right? These are the type of channel protein. So channel protein, they basically transport ion, right? So those who transport ions, they are basically known as channel protein, right? When channel protein, any channel protein, they transport just one substance in one time, these are called uniport. So they transport the one substance in one time, right? Either inside or outside. They are called uniport. So those channel protein which transport only one substance, they are called uniport. Those channel protein which transport simultaneously two substances, they are called co-transport. Again, they can be of two types. If channel protein is transporting both of the substance in same direction in one time, this is called symport. If channel protein transporting two substances both in opposite direction, right? So this is called antiport, right? So channel protein can be uniport, symport, or antiport. Is it clear? Here I have told you that sodium and hydrogen, they are the uniport, and fructose and sodium, they are symport, right? In digestion, we study in digestion, right? Chloride and like this thing we have studied in digestion, right? This part we have studied in digestive system, digestion. This part we have studied in, right? This is the right. Is it clear? Uniport, symport, antiport. This will be asked in your school exam. Active diffuse, active transport, passive transport, facilitated diffusion. They'll ask you two marker question, smallest question in your school exam, right? So now, are you aware with these terms? Impermeable, permeability, semi-permeability, selective permeability, or differentially permeable?
Yes, sir.
All of you, all of you, Shafullah,
So
Saeed.
semi-permeable, sir.
Okay, look, impermeable, those which are not permeable for anything, nothing can cross through that membrane. Permeable means something can cross through this membrane. Semi-permeable means like a particular size or particular type of the substance, particular size of the substance can cross through the membrane, like water. So membranes are like permeable for water only. So particular size of the molecule can be passed through the membrane, right? But in selectively permeable or differentially permeable, only particular type of the substance will pass when it is required. Like plasma membrane is semi-permeable for water, right? Cell wall is permeable for water, right? Plasma membrane semi-permeable for water, but mitochondrial membrane is selectively permeable. It will allow only certain type of the substance to cross through it and when it is required. Is it clear? Clear?
Yes, sir.
Yes, sir.
Okay. So let's move in the next part. So next heading is osmosis. Now let me explain you what is the osmosis. Look. So before going into osmosis, I would like to tell you what is the, just first let me tell you what is the osmosis. Look, these are the two chamber, right? So this is the first chamber and this one is the second. So between these two chamber, right? Between these two chamber, the transport of substances taking place. So what we did, suppose that this is the there is the this is the water molecule. This is the just pure water molecule, right? So in the chamber number one, so let me put the number. Chamber number one. In chamber number one, there is a pure water, and in chamber number two, there is water and sucrose. And between these two, there is a membrane called semi-permeable or differentially permeable membrane, which allow a certain type of the substance. So with the help of this membrane, only water molecule can pass, but the sugar molecule cannot pass, or solute molecule cannot pass. This sucrose molecule cannot pass, right? So what will happen? Water molecule concentration is higher over here. or in number 1. So in number 1 channel, water molecules are more. In number 2 channel, water molecules are less. So water molecule will start moving from their higher concentration to lower concentration. First line. So high concentration water molecule will move from its high concentration to low concentration. Then this is the low concentrated solution. So low concentrated solutions are called hypotonic, and higher concentrated solution called hypertonic. So we can also say the same thing. We can say that movement of water molecule takes place through from hypotonic to hypertonic solution, right? From hypotonic solution to hypertonic solution, right? Again, the movement of water molecule takes place from low concentrated solution to high concentrated solution. This is called osmosis through the semipermeable. It always takes place through the semipermeable, right? And this movement of water molecule will keep on going until at the both of the end concentration become equal. That is called equilibrium situation. When they will achieve the equilibrium situation, that is the concentration at the both of the end will become equal, then the process will stop and become stabilized. Clear? Tell me, osmosis clear or not? There are 3 definitions which I have told you over here. Please look carefully at the screen and let me know.
Sir, can you explain hypertonic and hypotonic again?
Okay, right. Look, hypotonic means low concentrated solution, right? Where concentration is low, and hypertonic is more concentrated, right? Just for sake of example, let me tell you here separate. Hypotonic. So what is that? Suppose that I have a tumbler. In this tumbler, I have 1000 milliliter of water, right? So here I have 1000 ml water, and then I have 50 gram of salt, right? And comparatively, right? So here in this solution, the same way I have 1000 ml of water, and here I have 100 milligram of salt. So this is the higher concentration. So what I will say, I will call it hypertonic, right? So where the concentration is low, so low concentrated solution are called hypotonic. Low concentrated solution, right? Low concentrated solution are called hypotonic, and high concentrated solution are called hypertonic. Is it clear?
Yes, sir.
So look at once more, and in case if you have any doubt, please ask. Osmosis is important for a school examination. Very important. School teacher generally ask with the diagram, and even you have to put the diagram as well. Look, first definition in this is in the term of water. Second definition is in the term of solution. Third definition is also in the term of solution. That is the difference, because they do ask question in all 3 ways in NEET exam. Clear?
Yes, sir.
Look, now this is a cell. Now I want to tell you what is the DP, diffusion pressure, and DPD, diffusion pressure deficient, right? So first I will tell you what is the diffusion pressure. Pure water have highest diffusion pressure, right? And if we make solution, means if we will add solute, then diffusion pressure will decrease. So what is the diffusion pressure? Diffusion pressure is actually the kinetic energy of water molecule or free energy of water molecule, right? Diffusion pressure is just right? Free energy of water molecule, right? So you can understand here the free energy is more and here the free energy in number 2 free energy is less. So movement will take place from where? From higher free energy to lower free energy, means from higher diffusion pressure to lower diffusion pressure. Is it clear? So suppose that here diffusion pressure is 10 mmHg water and here diffusion pressure is 4 mmHg. Now what will happen? Now the water will start moving from higher diffusion pressure to lower diffusion pressure until unless diffusion pressure becomes equal at both of the ends, 7 mmHg. When diffusion pressure will become equal at both of the ends, that is a 7 mmHg, then the diffusion will stop. This is called diffusion pressure. Clear?
Yes, sir.
Fine. One more. What is DPD? Diffusion pressure deficit, or this is also known as suction pressure. Right? The second heading. DPD, diffusion pressure deficit. Keep on with your notes with along with you. What is a diffusion pressure deficit? Look, in the number 1 tumbler number 1, the diffusion pressure was 10 mmHg. In the number 4, diffusion pressure is 4 mmHg. Now to bring at the equilibrium. So when they will become at the equilibrium, the diffusion pressure must be 7, right? When they will achieve this 7 at the both of them, diffusion will stop. So how much deficit is over here in the number 2, tumbler number 2? In tumbler number 2, to get an equilibrium, the deficit is of only 3 millimeter Hg, mmHg, right? Am I clear? So when pressure will over here become 7, then diffusion will stop. So how much deficit is there? Means how much less is there? So there is only 3 mmHg is less over here. Yes? Is it clear or not?
Yes,
Are you
sir.
getting my point?
Yes, sir.
Yes,
So
sir.
3 mmHg is less over here. So this is called diffusion pressure deficit. Means deficiency of 3 is over here. So this is called DPD, diffusion pressure deficit. Right? So what is the diffusion pressure? Movement of water molecule takes place from higher energy to lower energy, higher free energy to lower free energy. So that is called diffusion pressure, right? This is the pressure which this molecule impart on the semi-permeable membrane. What is the diffusion pressure deficit? The amount of the diffusion pressure which is less at particular point is called diffusion pressure deficit, right? DPD. Now come to the concept of this. What is the osmotic pressure? What is the Turgor pressure? What is the wall pressure? Right. So, movement of water molecule takes place from higher to lower energy level, right? So whenever the osmosis takes place, so due to the osmosis, water enter inside the where vacuole. So the amount of the vacuolar sap in water amount increase in vacuolar sap, right? So due to osmotic pressure, water enter into vacuole. When water enter into vacuole, these water molecule they put pressure in the plasma membrane in this direction. This is called osmotic pressure. So osmotic pressure is the pressure which is imparted by molecule on semi-permeable membrane during the process of osmosis. That is called osmotic pressure. So osmotic pressure is taking place towards the inwards in the cell. What happened? Whenever the water will enter inside the cell, then what will happen? Cell will swell and this water will start putting the pressure outside, right? So this is called cell will become swell, means cell will become turgid. Swell means turgid. So this is called Turgor pressure, and Turgor pressure takes place toward the outside, right? So osmotic pressure toward the inside and Turgor pressure outside. When this vacuole or this water molecule will push the cell wall outside, then the cell wall will also push it back, right? So that is called wall pressure. So the pressure which is imparted by cell wall inside is wall pressure. The pressure which is imparted by water molecule outside, that is called Turgor pressure. And wall pressure is always equal to Turgor pressure. WP is always equal to TP, right? Clear?
Yes, sir.
So look, there is a formula: diffusion pressure deficit, DPD, right? DPD is diffusion pressure deficit. And diffusion pressure deficit, DPD. So the formula is DPD is equal to osmotic pressure minus wall pressure. And wall pressure and turgor pressure are equal. So here, in place of wall pressure, I have put over here. We can also say DPD is equal to osmotic pressure minus turgor pressure, right? So tell me, go through with it and tell me, is it clear or not? And if not clear, then I'll tell you again.
So what is DPD?
The deficiency of pressure at another end. Look, suppose that I have 5 rupees and you have 10 rupees, right? So means you have 10 coin and I have 5 coin, right? So where the money is more? At your end, right? And what is the rule? We must have equal amount of the money. So to equalize money, how much you will give to me? 3.5 coin? Yes, Yusra, or not?
Yes, sir.
Then we both will have 7.5, right? So how much deficient to me? How much deficiency is there to me? 3.5.
Yes, sir.
That is the DPD. So when money will come from your end to me, that is the diffusion, right? And which is deficient with me, that is called DPD, right? Now you can understand in the same way of water. So suppose that what I have told you, what is the DPD? Look here, the this is the pure water, right? And osmotic pressure is 6 mmHg, right? And here there is the solution and this is the 4 mmHg, right? So 6 mmHg and 4 mmHg. What will happen? The water molecule will start moving towards from this end to that end, right? And what should be? There should be 5 at both of the end, right? To become equal. So how much deficiency is over here? What is the DPD? 1 mmHg. Is it clear? Deficiency is of 1 mmHg. Clear?
Yes, sir.
So DPT is the pressure deficit, means the pressure is less at the second end. That is called DPT.
Okay, sir. Thank you, sir.
Welcome. Right. So Saeed, clear? Is it clear, Saeed?
Yes, sir.
Okay. So let's move in the next. Actually, these are the very complex concept. And if you get concept once, you will get always. So feel frankly to ask me if you have any doubt, right? Now come to the next. So what is a water potential? The free energy of water is called water potential, and we this is the free energy, means chemical energy. So pure water have maximum free energy, right, or maximum chemical energy, and we call it water potential, right? So pure water have maximum water potential. So maximum free energy of water or free chemical energy of water is called water potential, and we represent it with the term that is called psi W. See, this is the psi W, right? This is the psi W, right? So psi W is water potential, right? So pure water have maximum potential, and maximum potential of water can be 0, right? So what is the maximum potential energy of water? That is 0, right? And whenever we add a solute to the pure water, then the potential energy or free energy of water decreases. So if it will decrease, so what will be the solution energy of solution? Free energy of solution less than 0 or not? Yes, I said that the water potential is maximum free energy of water molecule, or we can say this is the chemical energy of water molecule, right? So that is called water potential. And for a pure water, water potential is 0, right? So what will happen? What will happen when we will add solute to it? Suppose that in pure water I have added glucose. So what will happen? The energy of the water will decrease, free energy will decrease. So water potential will decrease. If it will decrease from the 0, what will happen? It will become negative. Is it clear or not?
Yes, sir.
So a pure water always have 0 potential energy or 0 water potential, and a solution always have negative water potential, right? So what is the osmosis? Movement of water molecule from higher water potential to lower water potential is called osmosis. This is the modern concept, right? So look, here this is the pure water, and here this is the solute addition. So suppose, what is the solute addition? So whenever the energy, suppose that this solution have the energy -2. So how does it -2 came, right? Addition of solute. So the amount of water potential decrease by addition of solute is called solute potential. Is it clear? The maximum free energy of water is called water potential. When we add to solute, the amount of energy or free energy decreases, water energy decreases or water potential decreases. We call it solute potential, right? Now there is a formula. Water potential is equal to solute potential plus solute potential. Solute potential is represented by psi s, and water potential psi p, psi w, and pressure potential psi p, right? So there is a formula. Water potential is equal to solute potential plus pressure potential. What is the pressure potential? Pressure potential is that amount of the pressure which decreases by addition of the or we can say the change in the pressure of water by addition of the any solute to it. So what is the water potential? Yusra, what is the water potential? The amount of free energy available to water is called water potential.
I can please repeat that once more.
I'll look again. Look, look, water potential. Suppose that let me tell you what is the water potential. Look, this is the, suppose that there is a free water, right? Pure water. So pure water molecule, the water molecule move here and there, and collide, right? They have maximum free energy. They have maximum free energy, right? Pure water have maximum free energy, right? And when we add solute to it, right, their energy decreases. Why? These free water molecule collide with solute molecule, right? Clear? Look here in this diagram. You can see over in this diagram, right? Here the water molecules are more, so they have more free energy. They can move here and there. So this is called water potential. Water potential is free energy of water molecule, right? Here the solid molecule, we have added more solid molecule. Now the water molecule will collide more, right? So what will happen? The energy of water molecule will decrease. Is it clear or not? Right?
Yes, sir.
So this symbol is called psi. This is called psi, right? And psi w is a symbol which represent water potential. Now what is water potential? This is the maximum free energy of water molecule. right? This is the maximum free energy of water molecule. Means this is the water potential is the free energy of pure water molecule, right? So what is the pure energy of water molecule, or what is the energy of pure water molecule? Zero. Now what happened? So pure water have water potential zero. Clear? Pure water have a water potential zero. So when I will add some impurity to it, when I will add some solute molecule to it, what will happen? The energy of what will happen? The energy of this water molecule will decrease, right? So that we call solute potential. And suppose that energy decreases by -2, right? So this is called solute potential. Is it clear? So suppose
Yes,
that
sir.
if you have 1 glass pure water, this have the maximum energy of water molecule, right? And what you call it size psi w, and that is the 0. Now what happened? Your sister came and it put one spoon sugar to it. Now what will happen? Those sugar molecule will start colliding with water molecule and they will decrease the energy of water molecule. What you call that decrease is in energy? Solute potential.
Solute
Why?
potential.
Because the energy of water molecule decreased due to the solute. Is it clear?
Yes, sir.
One more impact will take place over there. What? That is the diffusion pressure. Diffusion pressure of water molecule will also decrease or increase. What will happen? Whatever. So change in the pressure will diffusion pressure will take place. What we call that? Psi. And psi is pressure potential. Is it clear? And now we have derived a formula. What is water potential is equal to solute potential and plus pressure potential. What is that? That water potential is equal to change in the solute potential and change in the what? Pressure potential. Right? Now is it clear?
Yes, sir.
So movement of water or molecule always takes place from higher water potential to lower water potential. For example, here water potential is -2 this side, and this side water potential is -3. What is the maximum water potential? 0. So solution will always have water potential less than 0. Means the water potential of any solution will always be in negative. So movement of water molecule will take place from less negative to more negative. That is from higher free energy to lower free energy. Clear? Now did you got the water potential? Actually,
Yes, sir.
this is the osmosis, and we are representing the osmosis in terms of osmosis is a definition from chemistry, and now we are defining with the angle of physics. That is the only difference in osmosis and water potential. Both are the same thing. Look, let me tell you the same thing. I said that movement of water molecule takes place from higher energy to lower free energy. Means from higher water potential to lower water potential. Let me tell you in osmosis. Look, what is happening over here? What is happening? The movement of water molecule will take place from higher free energy to lower free energy. Yes, is it taking place or not? We are representing the same thing in terms of physics, in terms of free energy. Yes. Now, are you getting my point? So there is nothing difference between the osmosis and water potential. In osmosis, we say movement of water molecule takes place from hypotonic to hypertonic, or high concentration of water to low concentration of water. And in water potential, we say movement of water molecule takes place from higher free energy to lower free energy. We are repeating the same thing in different way. Clear?
Yes, sir.
Right. Now the stages of plasmolysis. What is the plasmolysis? Right. look, what is the plasmolysis? These are the stages of plasmolysis, right? So first, what is the plasmolysis? Simply, I want to explain you. Plasmolysis is the process by which any cell loses its water outside, right? So what happens? Suppose that I have a concentrated solution of salt. I have a grape. I put the grape in concentrated solution of salt. What will happen? The grape have low concentration and outside solution is more concentrated. Then water or juice of the grape will start coming off from itself and grape will shrink, right? That grape will shrink. That is movement of water molecule takes place from hypotonic to hypertonic, from grape to outside, and grape will shrink. And if in place of grape it is a cell, we call it plasmolysis, right? So what happened when we put a cell in a hypertonic solution? Hypertonic means high concentrated solution. What will happen? Water will start coming out from the vacuole. Means from hypotonic to hypertonic solution, water will start coming, and cell will become shrink. That is called flaccid state, and this is called plasmolysis. Right? Is it clear? This is called plasmolysis. Right? Done? Is it clear? Okay.
Yes, sir.
So when we put the cell more hypertonic solution or more concentrated solution, what will happen? More water will come out from the where? More water will come out from this central vacuole. This is the central vacuole. So more water will come out from the central vacuole, and the protoplasm will start leaving these spaces, right? Protoplasm will start leaving spaces at the corner, right? This is called second stage, that is the incipient plasmolysis, right? The first one was normal plasmolysis, or we can also call it limited plasmolysis, right? The second one is incipient plasmolysis. In incipient plasmolysis, look, in normal plasmolysis or limiting plasmolysis, protoplasm is not leaving cell wall anywhere. But in incipient plasmolysis, protoplasm is leaving cell wall at the corner. Now come to the third stage. If we put more hypertonic solution, now what will happen? That this is called evident plasmolysis. Here, the protoplasm will leave the cell wall everywhere except one or two places, like here. Look, like here, like here, look, like here, right? Like here. So these are the only three places where protoplasm is connected with cell wall. Our rest of the places it is leaving this space, right? And hypertonic solution enters in these space, right? So this is called evident plasmolysis, right? And this is the final plasmolysis. In final plasmolysis, what happened? Final plasmolysis, what happened? That protoplasm leave the cell wall, and in this is the complete space, right? In this complete space, hypertonic solution enter, and that is called final plasmolysis, right? After the final plasmolysis, if immediately cell is kept in a hypotonic solution or pure water, then cell can regain its original shape. Otherwise, it will permanently shrink or it may die permanently, right? So have a glance on this limited plasmolysis, incipient, evident, and final plasmolysis, right? And let me know is it if it's clear or not.
Yes, sir. Clear.
Yes. Okay. So when we after the plasmolysis, when we again keep these cells in pure water or hypotonic, when cell regain its shape, this is called deplasmolysis. So deplasmolysis is opposite of plasmolysis, right? Now the next concept, what is the imbibition? Can anybody tell me what is the imbibition? Imbibition. Tell me. What is imbibition? Look, simple. It's very simple. When any solid substance absorb the water, whenever any solid substance absorb the water, this is called imbibition. Like you take a seed, gram seed, whatever is sprouting, you want to sprout it, whatever the seeds you want to sprout it, what you happen? You soak this seed at the night time in the water, and at the morning you see the seed has absorbed water and their volume increased. And this process is called imbibition. Whenever a solid surface absorbs the water and increases its volume, this is called imbibition. Like soaking the seed. Clear imbibition?
Yes, sir.
Now the next concept is water absorption by plant. Look. So water is absorbed by the plant with the help of root. Root absorb the water from outside, right? How does the root absorb the water? Look, root absorb the water with the help of root hair, right? Root hair. So these root hair, they absorb the water. When they absorb the water, so water moves between the, so there are two paths of water, this pink color and this blue color. So whenever the water move, the space between the cells, that is called apoplastic pathway. So this red color pathway is apoplastic pathway. Whenever water do not enter inside the protoplasm or water do not enter inside the vacuoles. So suppose that if water do not enter inside the vacuole or cell wall or protoplasm, this is the called apoplastic pathway, right? So when water moves, the space between the cell wall, that is called apoplastic pathway, right? And whenever the water move inside the protoplasm or with the help of protoplasm and plasmodesmata, this pathway is called symplastic pathway. Clear?
Yes, sir.
Again, we have already studied the structure of root. Whenever we study the structure of root, the outer layer of the root is called epidermis. This is the outermost layer. This is called epidermis. And this root hair, these are the epidermal outgrowth, right? Inside the epidermis, there is a cortex. The last limit of cortex is endodermis. Endodermis have a particular kind of thickening. Endodermis have a particular kind of thickening. This is called Casparian strip. Casparium strip is impermeable to water. So the water cannot, in apoplastic pathway, water cannot pass through. So in Casparian strip, right, in the endodermis, there are a particular kind of the cells, and we call those cell passage cells. So water can pass through the passage cell. In endodermis, apoplastic pathway is not possible. Here, the water have to follow symplastic pathway, right? Means water have to come through the protoplasm. Water cannot come through the space between the cell walls, right? And then while crossing the endodermis, water reaches to the xylem. The same thing has been shown over here. Look, this green color lines, first one is apoplastic pathway. When water move, water do not enter in the epidermis, water do not enter, water need not to go to into protoplasm. It moves between the space between the protoplasm and cell wall. So this is called apoplastic. Even in cortex, the apoplastic pathway takes place, right? The movement takes place through apoplastic. But when it comes to the endodermis, apoplastic pathway is not possible. Here, the water move through the protoplasm, that is a symplastic pathway take place. Then in pericycle, symplastic pathway, and then it enters into the xylem, right? And now the symplastic pathway. And when the water enters through the protoplasm, right? So in epidermis, the symplastic pathway also takes place. The water moves through the plasmodesmata and again reaches to the cortex, symplastic pathway. When it reaches to the endodermis, symplastic pathway, again, right, it moves. So we can say in epidermal cell, symplastic and apoplastic, both of the water movement takes place. In cortex, symplastic, apoplastic, both movement takes place. But in endodermis, only symplastic, and in pericycle, only symplastic movement takes place. And in xylem, apoplastic movement only takes place. Is it clear? Symplastic, apoplastic pathway. And remember this diagram. This is important for your school examination. This diagram. It is also available in your NCERT as well. Tell me, guys.
Yes, sir.
Shahid, Shoaib, is it clear to both of you? Shahid, Mohiddin, Tell me. Is this concept clear to all of you? Mohiddin? Am I audible to you? Shahid? Not audible or not clear?
Yes, sir.
Okay, great. So this was the apoplastic and symplastic pathway. It's important for school examination. And this diagram is also important for school examination. Now, what is the mycorrhiza? So mycorrhiza is a special kind of the arrangement. So what happened? Like the larger plant, like the gymnosperms, they are they like pinus, like larger plant, like the pinus and like the cycas and all that. Their roots are weak water absorb. They are weak in water absorb. Angiosperms are very strong in concept of water absorb. That's why angiosperms are found in all kind of the habitat, everywhere. They are found in ice, they are found in desert, they are found in water, they are found in land, everywhere. But gymnosperms, they are weak in water absorption. That's why they are not found everywhere. They are mostly situated in mountainous area, right? So in gymnosperm root, what happens? Around the root, there are fungi. So fungi make a covering. This is the mantle of fungi. That is, mantle means that is the covering of fungi. So fungi make a covering around the root, covering of fungi. So fungi make covering around the root, and what it do? It provide water and mineral to the gymnosperm. In return, gymnosperm give it to the food. So both are helping each other. This kind of symbiotic relationship, where both are helping each other, then this kind of the symbiotic relationship is called mycorrhiza. So mycorrhiza is the association between the symbiotic association between the root of higher plant, right? So mycorrhiza is the symbiotic relationship between the root of higher plant like pinus, higher plant, and fungi mycelium, right? That is the call mycorrhiza. Is it clear?
Yes, sir.
What is the root pressure? So look, now what is the root pressure theory actually? Root pressure theory was, so there are number of the theories which explain how the movement of water takes place from root to shoot. So when I say or when I talk about the movement of water from root to shoot, then movement of water takes place even in 1 feet plant, 2 feet plant, 7 feet plant, and 120 feet plant or 300 feet plant of sequoia and eucalyptus, right? So the movement, how does this movement of water takes place? Number of the scientists came and they explained a theory, and there was the first scientist's name was Priestley. He is the same Priestley who have discovered oxygen, who have discovered that the oxygen evolved from the photosynthesis. So the same Priestley given root pressure. What he said that root absorb the water from the soil and they put the pressure in xylem, and with the help of that pressure, water move upward, right? So this theory is not effective for higher plant. Root pressure, yes, the root develop a pressure, but it's very low pressure. The pressure cannot permeable to movement of water in higher plant. So this is applicable for her, and root pressure develop only 1 to 2 bar. So this is not sufficient for movement or pushing the water in the higher plant, right? And root pressure takes place at the daytime, and it becomes absent at the time of drought or water scarcity. If water deficiency is there, water scarcity is there, even the root pressure will become zero, right? So the root pressure theory is a failed theory, and it do not properly explain the movement of water from root to shoot in higher plant. So this is the failed. Is it clear? Root pressure theory. Keep it in mind who explained the root pressure theory, right? Clear, guys? So you have to just keep this concept in your mind, because these three theories are not in your NCRT, right? Root pressure theory, vital force theory, theory of capillarity. They are not in your NCRT, but they are very important for your medical examination, right? So this is the root pressure theory. Did you got my point? Root pressure theory.
Yes, sir.
Okay. Again, there are a second scientist. His name was J.C. Bose, Jagdish Chandra Basu, right? So he given a vital force theory or pulsation theory. Just you have to remember the name of the theory, what he have given, and what actually is it was. Vital force theory or pulsation theory says that pulsation takes place in root cells, and due to pulsation, due to their vital force or living force, they pulsate and they create a pressure through which water move upward. This is completely wrong. No pulsation or no vital force is there in root cells, so that they could pulsate and they could create a pressure to movement, like pump. They could create a pressure so that the water move upward. So this was a failed theory, again failed, right? So vital force theory or pulsation theory, it is also a failed theory, and it could not explain how does the movement of water takes place in higher plant, right? Again, there is one more theory that is called theory of capillarity. It was given by Bohm, right? Bohm is also a very famous scientist. So Bohm said that in Xylem pipeline, they work as capillarity, and water moves upward with the help of capillarity. Of your physics experiment, you must have listened to capillarity. So movement of water takes place through the capillarity. This is also a failed theory. So these all three theory fail to explain how does the movement takes place in uh, higher plant from root to shoot. How does the movement of water takes place? Tell me, is it clear? All these three theory clear or not? That's still it. Done? Is it clear?
Yes,
All
sir.
these three theory. Okay. Now, if all these three theory fail to explain how the movement of water takes place, then what is the actual theory? So first, before going into that, how does the water movement takes place? First, try to understand what is the transpiration, or what is the transpiration pull. Look, water is transpiration. Why the transport is a transpiration? So water molecule moves out from the stomata, small pores, those are called stomata. So water movement takes place from them, and this unused water evaporate from the leaf surface. That is called transpiration. And why does the transpiration takes place? Can anybody tell me why the transpiration takes place? What is the purpose behind transpiration? And we call it necessary evil. Transpiration is called necessary evil. Why? Any one of you, tell me, please. Even I have written over here why the transpiration is called necessary evil. Tell me, yes, guys. Very simple. Transpiration takes place to cool down the body temperature of water, to plant. Like the sweating cool down our body temperature, right? Whenever the sweating takes place in our body, then the body temperature decreases or cools down, right? So this is called, this is transpiration. So whenever the unused water evaporate from the leaf surface, this is called transpiration. And why transpiration takes place? Water is very valuable to the plant. Still, plant loses 90% of its water. So the water which plant absorb out of 100%, they lose it 10% in transpiration. Only 2.2% water is used in photosynthesis, right? So why, if water is that much available, they they are hard earned water, they loses 90% water. So why? Because they have to cool down their body. They do not have fan, they do not have AC like us to cool down their body temperature. They always remains in exposed part. So plant transpiration takes place to cool down and maintain the body temperature of plant, right? That's why transpiration takes place, right? Transpiration is necessary, will, yes. Why transpiration is necessary, will, sometime school authority or school examination board ask this question. It's very important. It is necessary because if the body temperature will not be low, then what will happen? Then plant will die. If plant will not cool, it will die. Why it's evil? Because plant absorb water at like this is their hard earned substance, water, right? So plant spend lots of energy to absorb, lots of energy, lots of the processes to absorb the water, but they are compelled to, like, this is, they have to lose the water to cool down their body. So that's why it is evil, because they lose 90% of their water. That's why evil and necessary, because they have to cool down their body temperature. Now, types of transpiration. So first transpiration is stomatal transpiration. So stomata are a small pore-like structure, right? These are the stomata, right? They are found at the epidermal part. So 50 to 97% of the water is lost by the plant through stomata. This is called stomatal transpiration. Then cuticular transpiration. 3 to 10% of water is lost by cuticle or epidermal cell. This is called cuticular transpiration. And cuticular transpiration takes place in day and night, right? Now, the third one is lenticular transpiration or lenticellular transpiration. In lenticular transpiration, the transpiration takes place through the lenticels. So lenticels are found in the stem. So lenticels are a small pore-like structure. They are found in plant stem, and plant keep on losing the water from them, and this is called lenticular transpiration, right? So 0.1% water is transported through the lenticels, right? And then it keep on going on the day and night at both of the time, right? So that is called lenticular transpiration. So lenticels are found in the stem. Keep it in mind, these are the small pores found in the bark, right? 0.1% water is lost, so very small amount of the water is lost through them. Now then, to come to the bark transpiration. So there is a cracky covering of a stem. Like you must have seen a small mango tree, large mango tree. They have a cracks in their bark. So this cracky covering of a stem, they keep on losing the water day and night. This is called bark transpiration, right? So now I have written over here one more question in the form of note, that number of stomata, they are 1000 to 60,000 per centimeter square, or 10 to 600 per millimeter square. The number of stomata have been asked in medical exams. In case if you have not noted down, please note it down, number of stomata, right? So is it types of transpiration? Is it clear? Stomatal transpiration, cuticular
sir.
transpiration, lenticular and bark transpiration. Is it clear? Okay. So did you note it down, number of stomata? I think you must have noted down. Number of stomata. This have been asked in medical examination. Number of stomata. It is not in there in NCERT, but they have asked this question. Right? Now, the below diagram that is showing the structure of stomata. The diagram can be asked in your school examination. So,
Sir, can you please show the number of stomata on screen?
yes, number of stomata there 1000 to 60,000 per centimeter square or 10 to 600 per millimeter square. And one more thing I want to tell you, stomata are more abundant in lower surface and less in upper surface. Very less in upper surface.
Done, sir. Thank you.
Okay. Right. So now this is the structure of stomata. Right. So look, stomata is made up of two cells. Right. So these are called guard cells. They are kidney-shaped cells, and they have inner wall is thick wall. And look, their inner wall is thick wall, and their outer wall is thin wall. Right? They have outer thin wall and inner thick wall, and they have vacuoles. Right? And these are kidney-shaped cells, and they are called stomatal cells. Between these kidney-shaped cell, there is a opening that is called stomatal opening. Right? That is called stomatal opening, right? So this is stomatal opening, right? This is called pore. Around these guard cells, so there are guard cell. Around the guard cell, there are some normal cells. These are basically epidermal cells. These are called subsidiary cells, right? So water moves from subsidiary cell to guard cell, and then a guard cell loses the water through the help of stomatal opening. When they loses the water, they become flaccid and they closes down, right? So when water comes inside the guard cell, they swell, stomata open. When they lose the water, the cell become flaccid or shrink, and due to shrinkage of cell, stomata closes, pore closes down, right? That's how the stomata opening and closing takes place. Clear?
Yes, sir.
Okay. This is the diagram of leaf. Look, I have told you that the lower surface, the like upper surface, is made up of a cuticle. So what is a cuticle? Cuticle is a waxy layer, right? Cuticle is made up of a waxy layer, right? So from this waxy layer, this is the water resistant, right? So movement of water or transpiration do not take place to the upper surface. They have very less stomata, but between the and below the cuticle there is a upper epidermal layer. Then these palisade layer, mesophyll cell, so they are responsible for photosynthesis. They are responsible for photosynthesis, right? And this is spongy parenchyma, and they have lower epidermis, lower cuticle, right? And like lower surface, they have small cells for opening. They are called stomata, right? And they have guard cells and all that, right? Clear this all concept, transpiration, concept of transpiration, concept of stomata, types of transpiration. Is it clear to all of you? Yushra, Rafi,
Yes, sir.
Moinuddin, Shafullah, is it clear to all of you? All point.
Yes, sir.
So I should move in the next topic. Now, how the transport of the water takes place. So we have studied there are failed theory, right? So failed theory we studied, that is there were the root pressure theory, vital force theory, and all these theories. So they were the failed theory. Then we studied what is the transpiration. Then now there is a theory which explain the movement of water in all the plants, right? From root to the shoot, right? This is called actually the transpiration pull theory. Now, what is the transpiration theory? Or this is called cohesion and tension theory, right? Look, cohesion and tension theory, or we call it transpiration pull theory, right? This is the theory which actually nowadays explain how does the movement of water takes place. Look, transpiration pull theory explain that when the transpiration takes place through the stomata, then they create a pull force. They pull the water column like a rope. They pull the water column like rope. That is called pull, right? Transpiration pull. And the second thing, root pressure is creating a pressure, right? Now, what is the cohesion and tension? Look, pull force is created by transpiration. That is for sure. Now xylem, this is the xylem. Look, this is the xylem. Xylem, inside the xylem, look, inside the xylem, there are some water molecules. So water molecule are attracting each other. Every water molecule attract another water molecule with the help of a force that is called cohesive force, right? And this is called water column, right? This is called water column. Look, this is called water column. So one water molecule is pulling another water molecule is pulling another one. So all the water molecules are pulling each other with the help of a cohesive force. That's how they make a string, or that's how they make like column of water, right? Right. So this is called cohesive force. Cohesive force is the force which pulls the molecule of same type. So the force between the same type of the molecule, or pulling force between the same type of the molecule, is called cohesive force. So one water molecule attract another water molecule with the help of a cohesive force. Like we have a cold drink bottle, we put a straw and we pull the water, like pull the that cold drink inside our mouth. So the force we create with the help of our mouth, suction force, that is called transpiration pull. And one water molecule is like attracting another water molecule. One water molecule is pulling another water molecule with help and making water column. This is called what? This is called cohesive force, right? So cohesive force makes a water column, right? Again, the water molecule and the xylem wall are of different material. So different molecule attract each other, or different substances attract each other with the help of adhesive force. So what happened? The cohesive force give a tensile strength, right? Give a tension and make a water molecule column, and adhesive force do not allow this water molecule to fall back due to gravity, right? And this is pulled by with the help of transpiration. This is called cohesion or tension theory, or for transpiration pull theory. So, like we are pulling the cold drink with the help of a straw. So, straw is xylem pipe. The pulling force is like transpiration, and water column is made up of cohesive and adhesive force. That's how the transpiration pull theory explained. That's how the movement of water molecule takes place. Even the highest plant of 112 meters, Sequoia sempervirens or Eucalyptus rhagnus, that high means it can. And this is the sufficient pressure to look. This theory, cohesion and tension theory, it was given by Dixon and Joly in 1894, and this is also called ascent of sap. And cohesion, adhesion, and surface tension, they keep the water in their place. And water column can bear a tensile strength of 100 atmosphere up to 1 atmospheric pressure can pull the water up to 10 meter. 10 to 20 atmospheric pressure can pull the water up to 130 meter, right? Which is sufficient enough to reach the water in the highest part, at the top of the highest plant of the world. Tell me, see the theory, have a glance on the theory, and if you have any doubt, all of you, please ask me. This is the theory which explains how the movement of water molecule takes place. Tell me, guys.
And it's
Clear? Mohiddin, is it clear? Shoaib and Shafullah, clear? Shafullah, transpiration pull theory.
Yes, sir.
Okay, fine. So I think we should move in the next theory, right? Now come to the next. Transpiration, photosynthesis, a compromise, right? Look. So transpiration pull, we have understood. Photosynthesis, that is the, we know that photosynthesis takes place the food formation process. Transport of mineral, right? So transpiration pull is responsible for pulling the water, right? And water is required for photosynthesis, right? Transport of mineral takes place through transpiration, right? Transpiration decreases the temperature of plant by 10 to 15 degrees centigrade, right? They maintain the shape and structure of the cell, right? Water maintain. And uptake of mineral, most of the minerals are taken through the active absorption, means the minerals are taken with the help of water. They follow the same path which is followed by the water, but yes, the most of the minerals are taken by the plant at the expense of ATP. So ATP is spent. So now the chief sink for mineral, right? That is the growing. That is the chief sink, means that is the main part where the minerals are required. So that is the growing region or meristematic region, right? And look, what happened? That phosphorus, silver, phosphorus, sulfur, nitrogen, and potassium. So these all are the substances. They are remobilized. So what is the remobilizing? Whenever a death of a leaf takes place. So death means senescence of leaf. So senescence means the leaf is about to fall or moving towards the death, progression towards the death. Progression towards death. Progression towards death, right? So whenever the senescence of leaf takes place, so they take back the important mineral and substances. So the mineral or substances from the older leaves, they move back to the main plant, and this is called remobilization. So during the process of remobilization, phosphorus, sulfur, nitrogen, and potassium, these before the leaf fall, they revert back or give back these phosphorus, sulfur, nitrogen, and potassium to the plant. But calcium is not remobilized. Why? Because it is component of structural plant, structural part, structural part, right? Tell me, remobilization or uptake of mineral is clear? Keep it in
Yes,
mind,
sir.
this is the active process. Okay, fine. And always remember this question, calcium ion is not remobilized. So those ions which are remobilized, this question and this question, both of the question have been asked in medical examination many times. Now the transport of food in phloem. This is very important. It is called mass flow hypothesis or pressure flow hypothesis. Why pressure flow hypothesis? Because movement of food takes place from higher diffusion pressure to lower diffusion pressure. First thing. Why mass flow hypothesis? Because mass is transferring, that is, food is transferred from one point to another point. So pressure flow hypothesis or mass flow hypothesis explain how does the movement of food or organic substance takes place with the help of phloem. While transpiration pull theory tell how the movement of water takes place in xylem from root to shoot. Pressure flow theory or mass flow theory tells how the transport of food takes place from higher to lower concentration. Look, xylem and phloem. Now you can see in the diagram, this is the xylem tube and this is the phloem tube, right? Please be attentive and keep your eye over here. In the process, these are the source. What are the source? Source are those parts from where the formation of food takes place. One more thing I want to tell you. What is the source? So source are those part where food formation takes place. Food formation takes place. Now, what is the sink? Those part where food storage takes place. Where food storage takes place, right? So this is the source and the sink, right? Now, let me tell you how does the source and sink relation takes place. Here, in source are the leaves or source where the photosynthesis takes place. Formation of food takes place in the form of glucose, but glucose is the monomer or monosaccharide. So transport of food do not takes place in the form of glucose. First, glucose changes into, that means monosaccharide changes into disaccharide, right? So from glucose, formation of sucrose takes place. So first important question, in the food translocation in phloem or in transport in phloem, the transport takes place in the form of sucrose, not in the form of glucose. Transport of the food takes place in the form of sucrose, that is disaccharide, not in the form of glucose, which is monosaccharide. First important question for your medical exam. Again, from source cell or mesophyll cell, right, the food is transferred to nearby phloem. In phloem cell, first food is transported into companion cell. We know that companion cells are active cells, right? Companion cells are living cells. So food is transferred from, with the help of, to the process of diffusion, or we can say to the process of osmosis, right? So through the process of osmosis, food is transported in the form of sucrose from the source or leaf cells or mesophyll cells to the companion cell. Now companion cell collect a lot of the food. Companion cell transfer this food to nearby sieve tubes, right? Whenever the food is transferred from companion to sieve tube, this is the active process. That is expansion of energy takes place. Energy is spent over here. At this point of time, energy is spent. And the process of transfer of the food, the process of transfer of food from companion cell to phloem is called loading. Right? Companion cell is called loading. So in loading process, transfer of food takes place. Right? Whenever the food is transferred from companion to phloem, right, this is called loading. When food reaches to the sieve tube element, these sieve tube element, they have higher concentration of water. Hence, that's why they take the water from nearby xylem element by the process of osmosis. Movement of water molecule takes place from higher energy to lower energy, right? Means from higher osmotic pressure to lower osmotic pressure. So they take the water from nearby xylem. And by this way, movement of this food food in sieve tube takes place with the help through the concentration gradient or diffusion from one cell to another cell, from one cell to another cell to another cell. That's how the movement of substance takes place. When substance reaches to that place where the storage or unloading in take place, so what will happen? From phloem or from sieve tube element, the food is transferred to companion cell, nearby companion. Again, ATP is spent, means energy is spent. Again, this is the active process and this is called unloading. So look, transfer of food from companion to nearby sieve tube element is loading. Transfer of food from sieve tube to the companion cell at the sink is called unloading, right? After the unloading, in loading and unloading, both are the active process. At the both of the process, energy is spent over here, right? After that, the food is transferred from companion cell to root cell or sink cell, where food is converted into from sucrose to starch. Starch is a polysaccharide and it is insoluble. That's why starch detaches with the water, and again water comes back to nearby xylem and water recirculate and comes again its own pathway, right? So that's how the water takes the role of only picking the sucrose from one point and dropping at another point, right? And that's how the water comes back, and that's how the translocation of food takes place. And this concept, this theory is called mass flow hypothesis, or this is also called the pressure flow hypothesis. Tell me, guys, is it clear or not? Transport of food. Tell me, all of you.
Yes, sir.
Shahid, Yushra is very prompt. She always respond. And Shafullah, Shahid, and Shoaib. Tell me, is it clear or not? If not, then where? So this was all about the complete chapter. And before moving to the next chapter, please go through with your NCERT. I'm just giving you some time. Go through with your NCERT. And if you have any doubt in your NCERT, please ask me right now.
Sir, can you explain electron transport chain?
Yeah, I'll explain. This is in next chapter, next chapter. I'll tell you. Electron transportation. I'll tell you. In this chapter, go through with your NCERT in this chapter and let me know. I'll explain all of the chapter, all of the plant physiology chapter. Electron transport chain of photosynthesis or respiration?
Both, sir.
Both. Okay, I'll tell you.
Sir, can you take photosynthesis tomorrow?
Photosynthesis and like next after this chapter.
Yeah, sir.
Yeah, definitely after this chapter, I'll start with the photosynthesis. No issue. So when you guys have exam?
Sir, my bio exam is on Tuesday.
On this Tuesday, this coming Tuesday.
Yes, sir.
Okay, so ask me right now if you have any doubt in your biology examination in biology. And what about you, rest of the people? Said and Shakullah? Shahid, when you have your exam? Yusra, your exams are nearby. Just
Sir, 6th, sir. 6th of March.
6th of March. So no issue. Yusra, this is time. This is high time for you. So ask me any doubt if you have any doubt.
Yes, sir.
Fine. So next chapter, I'll pick the photosynthesis, right? And first, I'll tell you the electron transportation. Then I'll move ahead. So, what about you, Alex? Saeed have told me that he has exam in March. Shafullah, when you have your exam is
On 6th of March, sir.
6th
Final
of March.
exam.
Okay, fine. And the same with the Saeed, Moinuddin. Yes, same with you. 6th of March onward, right?
Yes.
Okay, fine. No issue. So next, start with the photosynthesis, right? So I'll skip the mineral nutrition chapter, and then I think mineral nutrition is out of your syllabus. Like it is out of yours. It's not in your syllabus. Yes.
Yes, it's not there.
It's not there. I have listened that. This is the photosynthesis, and photosynthesis first, as you have requested, then I'll tell you what is the electron transport chain. It's important as well. They can ask you. Yes, both of the electron transport chain and electron transport system, both are important. So till the time, I'll tell you what is the electron transport chain. So look, what happened in electron? This is Z scheme. So why it called Z scheme? Because the energy pattern or energy graph looks like Z. That's why we call it Z scheme. And this is called non-cyclic photophosphorylation. Non-cyclic, why? Because electron ejected from one point and it is received at another point. It do not comes back. So this is non-cyclic. From one end, electron is leave the place, and another end it is received. Photophosphorylation, because synthesis of ATP takes place. That's why it is photophosphorylation. So what happened? Photolysis of water takes place, right? Water molecule breaks down, right? Photolysis of water takes place. Water molecule breaks down, right? And whenever the water molecule breaks down, hydrogen is released. When water molecule breaks down, the electrons are released. Actually, when 2s2, when two water molecule are from one molecule, water molecule, two electrons are ejected. So these electron reaches to photosystem second, and photosystem second is called P6 at T. Actually, it comes in the reaction first, but it has been discovered in later. That's why this is called PS2. So this is a sequence is on the basis of discovery. First PS1 have been discovered, then PS2 have been discovered. But in the electron transport chain, the role of PS2 comes first, right? So PS2 is called, it have a reaction center called P6 at T. These electrons get extra energy from the photosystem and they jump outward. These electrons are received by a cytochrome called pheophytin. From pheophytin, they are received by plastocunine. From plastocunine, they are received by cytochrome B6F complex. Whenever the electron passes through cytochrome B6F complex, formation of ATP takes place from ADP. This is called photophosphorylation. That is, photophosphorylation means formation of ATP. Formation of ATP. So this is called photophosphorylation. So from cytochrome B6F complex, it reaches to plastocyanin. But whenever electron is passing through from one cytochrome to another cytochrome to another cytochrome, then what happened? During this process, energy is losing. That's why the graph is coming lower down, right? So again, the electron reaches to chlorophyll P700. So PS1. When electron reaches to PS1, again it give energy to electron. Electron again jumps upward. So again the graph is upward, graph jump. From P700, electron reaches to A0, a special chlorophyll complex, then FX, then ferredoxin, FD, and then finally electron is received by NADP plus and it convert into NADPH. So NADP plus is the thing, or this is the final electron receiver, which finally receive the electron, and that's how the non-cyclic photophosphorylation takes place, or non-cyclic electron transport chain takes place. So symbol Pheo is pheophytin, PQ is plastocunine, cytochrome B6 complex, cytochrome B6 complex, cyto B6F, PC is plastocyanin, FES is FES complex or FES complex, and FD is ferredoxin, right? Tell me, is it clear or not?
Yes, sir. Sir, can you please explain me Krebs cycle once?
Krebs cycle, yeah, yes. Shahid, is it clear to you? Saifullah, clear?
Yes, sir, clear.
Non-cyclic is more important. Definitely, I think they'll ask you non-cyclic, draw this non-cyclic electron transport chain or non-cyclic photophosphorylation, right, with the help of diagram. So you are asking, what do you ask? Krebs cycle. So, Krebs cycle respiration. Grapes. Grape. Yes. Look, this is Krebs cycle or TCA cycle. Krebs cycle is nothing but burning of glucose. Look, in Krebs cycle, look here, the Krebs cycle takes place in the matrix of mitochondria. So a 6-carbon compound glucose comes over here. Glucose changes into, during the glycolysis, glucose changes into pyruvate. Pyruvate is a 3-carbon compound, and this 3-carbon compound, when enter into Krebs cycle, what happened? The pyruvate cannot enter into Krebs cycle. So what happened? Formation of NAD to NADH2 takes place, means production of energy takes place. So I have already told you that 1 NADH2 is equal to 3 ATP. So from 2 pyruvate molecule, formation of 2 acetyl coenzyme A takes place. But at the same time, what happened? Carbon dioxide is removed. So from acetyl coenzyme A is 2 carbon compound, right? So 1 carbon dioxide is removed and formation of NADH2 takes place. Again, the pyruvate changes to acetyl coenzyme A. From acetyl coenzyme A, coenzyme A part get detached and it changes into citrate. Citrate is 6-carbon compound. From citrate, it changes into isocitrate. It is also 6-carbon compound. From isocitrate, formation of alpha ketoglutarate takes place, right? And formation of, again, energy formation takes place. Formation of NAD to NADH2 takes place, and that is equivalent to 3 ATP. And formation of isocitrate takes place into alpha ketoglutarate. Again, 1 carbon dioxide molecule is lost, and this reaction takes place in the presence of enzyme isocitrate dehydrogenase. Alpha ketoglutarate is 5-carbon compound. Why? Because this was a 6-carbon compound. Again, 1 carbon dioxide molecule is removed. So this is called decarboxylation. Alpha ketoglutarate is 5-carbon compound. Again, decarboxylation takes place. 1 more carbon dioxide molecule is lost. Formation of NAD to NADH takes place. NADH is equal to 3 ATP. This reaction takes place in presence of alpha ketoglutarate dehydrogenase. Again, the formation of succinyl coenzyme A takes place, which is a 4-carbon compound. From succinyl coenzyme A, formation of succinate takes place, which is also a 4-carbon compound. During this process, a GTP is produced. A GTP is equal to ATP. I have already told all this concept over here. So GTP is always equal to 1 GTP is equal to. So succinate, from succinate changes into fumarate. So from FAD to FADH2 takes place formation. Again, FADH is 1 FADH2 is equal to 2 ATP. From fumarate is 4-carbon compound. From fumarate to again malate takes place. Malate again changes in the presence of malate dehydrogenase. It changes into oxaloacetate. Oxaloacetate is 4-carbon compound. Again, this oxaloacetate, a 4-carbon compound, meet with acetyl coenzyme, a 2-carbon compound, and makes a citrate, that is a 6-carbon compound. That's how the process takes place and burning of glucose takes place, and everywhere the formation of carbon dioxide is removed and for the decarboxylation takes place and formation of energy takes place in the form of GTP, FADH2 and NADH. Here I have told you how much energy is produced. In glycolysis, 2 ATP and 2 NADH means 6 ATP are produced. In Krebs cycle, 2 ATP, 8 NADH2 and 2 FADH2, that is equivalent to 30 ATP takes place. 30 plus 8, total 38 ATP formation takes place. Tell me, is it clear, Yusra?
Yes, sir.
If you want, you can take the screenshot, but you must have, right? So this was something about
Sir?
the yes, tell me.
Sir, after citrate, sir, in the notes, sir, in the diagram, there are some like more compounds, sir. So what are those, sir?
Where? From
Like
citrate,
after citrate, sir.
isocitrate, this one, isocitrate, alpha ketoglutarate, all these concepts.
Sir, like from citrate, like
Wrong.
cis aconitis, some enzymes like that.
You need not to go with that, because that is not practically possible to remember, and you need not to. Actually, this is much more complex than your NCRT, right? This is sufficient actually. I have to, it is very complex table, a complex cycle, and that's why the most of the students do not remember it. Right? And the hormone I have written, just write it down, that those hormones, right? And please remember only these names which are there in this cycle. This is perfect. This is sufficient for both of your purpose, school as well as your NEET examination. Because the cycle which is given in your NCRT, that is less informative. It is not sufficient enough, right? So that won't work, but this will be like sufficient enough for you, right?
Yes, sir.
Otherwise, this is very complex cycle. It's really very difficult to remember, recall. Any more question?
Sir, can you please scroll down?
You can scroll. Okay, scroll up. And I will. Yes, now you can take this.
Okay, thank you, sir.
Welcome. So that's it for the day, and we'll see you in next class. And for you, like Moinuddin, and for you, like Saifullah, as you people have asked me for the electron transport chain of the both of these things. And so what I will do in the next class, I'll start with the photosynthesis. I'll try to complete photosynthesis, and the same time I'll tell you like this electron transport chain for the both of the respiration and photosynthesis part, right? And till that time, study, and let me know if in next class if you have any problem, any issue regarding any question or regarding any topic, right? And Yusra, all the best from my end, right? And uh,
Thank you, sir.
welcome, and come with the flying color with good marks, right? So yes, guys, that's it. See you in next class. And
Yes, sir.
come with more questions, right? So till the time,
Yes, sir.
bye-bye, good night, take care.
Thank you, sir.
Thank you, sir.
Welcome, bye-bye.
Thank you, sir.
Welcome.